Every ending is a new beginning, every conclusion a new direction. As knowledge transforms into action, we shape innovation with a reflection. Jai Hind, a very good morning to all respected dignitaries, distinguished speaker, esteemed faculty members, judges, researchers, scholars, and dear participants. It gives me immense pleasure to welcome you all to the day 3, the concluding day of the International Conference on Sustainable Development and Intelligent Innovation, ICSDII 2026. Over the past 2 days, we have witnessed insightful sessions, groundbreaking research, and meaningful academic exchange. Today, we gathered to continue this journey with renewed enthusiasm and conclude this prestigious conference on a note of inspiration and achievement. Today, we are honored with the presence of our esteemed speaker, Professor Dr. Preetam Singh, Principal Scientist, CSIR National Physical Laboratory, New Delhi, and Dr. Pravendra Kumar, Professor, Department of Bioscience and Bioengineering, IIT Roorkee. Before we proceed further with today's academic deliberation, let us begin the session with the felicitation ceremony of our esteemed invited speakers as a mark of respect and gratitude for their gracious presence and valuable contribution to this academic platform. It is now our honor to invite our respected Dean Sir and Dr. Shashiraj Tewatia Sir to kindly come forward and felicitate our distinguished speaker, Professor Dr. Preetam Singh, Principal Scientist CSIR, with a green planter as a token of our heartfelt appreciation. Please, sir. It is now our privilege to introduce the esteemed dignitaries who will guide today's session with their expertise. We are honored to have Dr. Lubhan Singh Sir, Additional Director Research, as the session chairperson, and we have Dr. Ashwini Kumar Sir, HOD Life Sciences, as a reporter. To honor our session chair, I would like to request Dr. Sarita Rana ma'am and Dr. Ritika Yadav ma'am to kindly come forward and honor the session chair, Dr. Lubhan Singh Sir, with a planter. Sir, please.
Without further delay, let us now formally commence technical session 8. And towards I invited lecture of the day. It is our privilege to introduce Professor Dr. Preetam Singh, Sir, Principal Scientist at CSIR, National Physical Laboratory, New Delhi. Sir completed his PhD in Experimental Condensed Matter Physics from IIT Roorkee and has extensive research experience in the development of thin films and nanostructured material for advanced gas sensing application. With over 60 scientific publications and significant contribution in the field of metal oxide and transition metal chalcogenate-based sensor, his work primarily focuses on detecting atmospheric pollutant using high-performance gas sensor devices. Sir has also served in prestigious roles including postdoctoral research in South Korea and continues to contribute actively to cutting-edge research in environmental sensing. Sir will be delivering his talk on the importance of national gas sensor testing and certification facility and metal oxide-based high-performance gas sensor. Please, sir, kindly come on stage and address the gathering.
A very good morning to all of you. First of all, I would like to give thanks for the organizing committee to give me the opportunity to deliver a talk. And also I would like to thanks to Madam for kind introduction. Mic is sitting. Today I will talk on today I will talk on that the importance. The work of the CSIR National Physical Laboratory is, it is a national custodian of primary national standards and has the responsibility of realizing the units of physical measurement based on the international systems, SI units. That means all the 7 physical quantities, meter, kilogram, second, temperature, ampere, candela, mole, all the physical 7 primary standard are in NPL. That means if you want to see the actual 1 kilogram or actual how the 1 second is measured and how 1 degree centigrade accurately measured, so all those standards are in NPL. In one country there is a one laboratory such kind of, that is called National Meteorology Institutes. So even not all the 7 basic units, all the derived units, which is generated based on the 7 units, we measure at CSIR National Physical Laboratories. So it is a signatory of the international body CIPM, which is authorized to issue the certificate. And once anything is calibrated from NPL, it is accepted worldwide for international trade. So it provides the apex level calibration and dissemination of standards for maintaining the traceability of measurement following the quality of systems. And from May 2019, now all the basic units measure in terms of physical constant, like in terms of Planck constant, Boltzmann constant, that kind of. So it is a transition from the physical quantity to the basic constants, and it is a traceability pyramid. Anything is measured from there, it is traceable back to the SI unit. That means SI unit measured by primary standards, then we generate second standard, and that is a reference standard which we provide to the firms or companies, and based on reference they generate their working standard and then measurement instrument. So the uncertainty is included at each point. So it is basically a uncertainty pyramid which started from the SI units. Okay, so now I will discuss on the current project on which we are working, National Center for Gas Sensor Testing and Certification, in collaboration of that funded by Ministry of Electronics and Information Technology and the CSIR, because gas sensor is highly required in today's situation. For it is mostly imported, but now there are lots of manufacturers and developer in India also. But as I told you, without certification and validation, it cannot say that how much it is accurate, or you cannot market it till then it is certified. Okay, so on that, for example, in recently, if you see the sensor market and the requirement and consumption, it is in the billions or millions. So there is a huge market for that. But in India, we need reliable certification facility so that we can also compete in the international market or international level. So it is just a data. If you see 2013 to 2019, actually it is expected from 1701 million to 2773 million. Okay. So the objective of this facility is to establish a state-of-the-art gas sensor testing and certification facility under quality management system, which will provide a complete and cost-effective solution of testing and certification to the Indian as well as manufacturer of gas sensor. Currently, for the measurement of gas sensor and certification, US and European standards are being followed for testing and certification, and Indian standard yet to be formulated. So this is the first step in this direction. Once this facility will be developed, then we will develop our own Indian standard to certify or calibrate these gas sensors. So initially this we will develop the testing documentation certification of various gases, including hydrogen, oxygen, and the air pollutant gases, including VOCs, volatile organic compound. It is a long list, because all the chemicals also in chemical factories, hospitals, there are on medicine, it is highly required that VOC sensor should also be calibrated. And why we can do that? For the periodic calibration, you need different standard also to calibrate this periodically. So we already have in NPL the other standard like traceable gases, temperature standard, humidity standard, electrical standards. So that is easy for us to access those facilities periodically to validate these facilities. So we will develop the protocol for testing and certification of these gas sensor and documents under with accuracy and precision with lowest possible uncertainty as per the standard of related to the environment for protection. And also it is to generate the awareness about the necessity of gas sensor testing and certification, because currently if you send any sensor to foreign, it taking the time also on cost also. Roughly what we have asked for the companies who have calibrated from the, you can calibrate from any NMI like NIST USA, PTV Germany, NMI Australia, this kind of. So they charges for one gas sensor minimum 50 lakhs, and it also takes time. So if we can do that facility within India, it will be a very good and boost for the sustainable development in India itself in this direction. So these are the slide change. So okay. So these are the different type of gases, hydrogen, CO2, NO2. For that, we are developing that facility. So it will help and boost up our startups, which is developed in India, because already many startups, MSMEs has contacted us for that calibration and certification facility. So it will boost our Indian manufacturers and MSMEs once this facility will be developed. So why these gas sensors are important? Because it is used for the air quality monitoring, environmental protection, industrial safety, healthcare, indoor safety, semiconductor and electronics, and hydrogen infrastructure, because hydrogen mission is also going on a very rapidly. So why the certification is required and what is the problem? Some I will discuss briefly. The uncalibrated gas sensor will be a lack of fit, linearity, zero and span drift, repeatability, lifespan, that means ki stability or the degradation rate there, selectivity. Why select? At a time one sensor can measure only one gas or it is measure multiple gases. So that cross sensitivity, why is so important? The selectivity, that I will discuss in later slide. The impact of humidity, impact of gas flow, temperature, uncertainty, because in India, if you see from one end to other end, there is a large variation in the climatic conditions. So it will not work same in all the conditions. So what we are developing, if we can simulate the environmental condition in that chamber as per the requirement of different locations. So to develop that facility, we will follow certain standards using that ISO standards for lab competence, gas reference mixing, analyzer testing, uncertainty evaluation, and then air quality certifications. So in short, that gas, what we have given the name acronym for that facility is gas tech facility. So gas tech facility under the quality management systems will ensure accuracy and reliability of gas sensor performance with the lowest measurement uncertainties. Okay, so in next part now we will discuss about the air pollution and the metal oxide gas sensor. So why air pollution is so dangerous for the environment and the human health? It is because it is one of the major challenge of 21st century. As per the report of WHO, 90% of world population lives in the region where air pollution levels are higher than the prescribed specified limits. So it is a global drive to tackle these problems. These are the major air pollutant components: particulate matters, that means the dust particles, and that gases. Yes, next slide. Why it is back? So it is a invisible killer. Air pollution may always not be visible, but it can be deadly. If you see, as per the report, 4.2 million deaths every year as a result of the exposure to ambient outdoor air pollution, and 3.8 million deaths every year as a result of household exposure to smoke from dirty cook stove and fuel. So that means 8 millions per year. So it's a huge number. It can causes of the various diseases. So that's why we need to measure the air pollution accurately. Then only we can make a policy or we can control the air pollution. This why it is no hand. Okay, so human body as a sensor. Okay, if you see our body work as a both physical sensor and chemical sensor. That means ki pattern recognition, sight, hearing, touch, smell, and taste. We can smell, we can taste, but sometimes we cannot taste at what quantity or what concentration those things is available. And some of these gases are colorless, tasteless, odorless. So that means you cannot taste or sense this. So that's why we need gas sensors or the other sensors to accurately measure the quantity and concentration of these gas sensors. Okay, so as I told ki, there are different threshold limit for gases. These are the safe limit for that environment is protected, or we can say, for example, for CO 35 ppm, NO2 3 ppm, SO2 2 ppm, and CO2 5000 ppm. If our sensor is not measuring accurately one gas or it shows the cross sensitivity, so that means if you are measuring some gases present 100 ppm. So if you see, if that is CO2, that is very safe because limit is 5000. But if 100 is available either SO2 or NO2, so that is highly dangerous, multiple times of higher than the prescribed limits. So that's why we need the accurate gas sensor which can measure the separate gas accurately, so that we can say it is below the threshold limit or higher on the. So there are various type of gas sensor which is mostly used for that. Optical sensor, which is compare the light intensity with the interaction of the material, metal oxide, that is measure change of the electrical resistance, and catalytic sensor, that is comparison of the electrical resistance, and the detection of electron transfer electrochemical sensors. So if you see the range of concentration on a large series of metal oxide gas sensor has the wide range, or all these metal oxide gas sensor can measure each gas which I have shown here. So mostly the established technology or the commercial technology which is well established right now also with the metal oxide gas sensor with some advantage or disadvantage. Okay, so broadly their principle is very simple. What is the principle? The metal oxide sensor is also known as chemi-resistive because it measures the change of resistance upon the absorption of the gas molecule on the surface of metal oxide. Every material, like it is mostly divided in p-type or n-type materials. That means at a particular temperature, like room temperature 100, 200, if we measure the resistance continuously with time, it will be a straight line. With temperature, the resistance will change. If you fix the temperature, you will get only the straight line of the resistance. But after exposure of these gases, the resistance will change. It can increase also, it can decrease also. That will depend on the what type of material we are using, n-type or p-type, and what type of gases we are detecting, reduced gas or oxidized gas. So depending on that, either resistance will increase or decrease, but you will feel the resistance change. So similar like metal oxide, we divide in two parts, n-type, p-type, and the gases we normally use as a reduced gas or oxidizing gas. So these gases can be measured with these. So for that, if we see, that is a basic simple principle. Oxygen is available here on every surface. Oxygen is absorbed on the surface due to its large electronegativity. They extract of the electron from the materials, causing the formation of oxygen ions. That means that oxygen is mostly present in form of oxygen ion on the surface. Depending on the surface temperature, it can be O2 minus, O minus, or O2 minus. So these are in some cases, this is a disadvantage for many materials. That means that those materials are not as a oxide, like selenite, sulfides, or some other. Then oxygen impurity will be there, material property will change, it will degrade, but it is a boost of metal oxide because they are already oxide. And what is their working principle on gas sensing? These presence of oxygen ion will beneficial for them. So we will take one example like CO gas. It is work on that, that oxygen ions will convert in CO2 and release an electron. So if it is a n-type material and after interacting with CO, it will have more electrons. So that means conductivity will increase and resistance will decrease. If it is a p-type material, then electron concentration will increase in p-type material. It will recombine with holes. So holes concentration will reduce in p-type. The resistance will increase. So that means if reduced gas will interact with n-type material, the resistance will decrease. And if it will interact or with the p-type material, the resistance will increase. Similarly, for oxidizing gas, to react that gases on the surface, it needs electrons. So if n-type material is there, it will extract the electron from the material. So the concentration will reduce, resistance will increase. And for p-type material, the concentration of hole will increase. So that means the curve will be vice versa. That means if n-type material is there, it is interacting with reducing gas, resistance will increase. And for oxidizing gas, the resistance will increase. So for p-type, it is vice versa. So there are some, we have developed our gas sensors. After that, how we will judge what property we need to look for gas sensor so that it is a good sensor or bad sensor. First is, so they are called sensing performance parameter. So first we take as a sensor response. That means we have two resistance, one in presence of air, that means without exposure any gas, and the other is Rg, where after interacting the material, the resistance will decrease. So their ratio will decide the sensor response. If resistance is decreasing, normally it is calculated in percentage. So we take delta R upon R, and when the resistance is increased, so if we convert in percentage, it is too high. So normally if you see the research paper, some give in the value of response in percentage or some give in the ratio. But in both cases, the resistance should be high. That means if sharp change will come in resistance, so sensor response will be high. Similarly, alag is selectivity. As I told, it is a property of a sensor from it is selectively detecting one gas among the mixture of the different gases, so that we can accurately define it is good for one. As I told, there are different threshold limit for separate sensor, so it should be selectivity should also be high. Similarly, sensitivity, it is the ability of a sensor to detect the analyte at even low concentration, that should also be high. Stability is the ability of sensor to provide result for a long time. It is used for one time, two times, or for 1 month, 2 months. That means ki stability should also be high, so that it can be used for long time. Response and recovery time. Response is once the gas is interacted with the sensor, how quickly the resistance is changed. So the total resistance value change, we calculate 90% of the total signal change. It should be fast. And similarly, once if we remove the gas, so that it can be reusable again, so recovery should also be fast. So sensor selectivity, stability, sensitivity should be high, and response recovery should be fast. So if we take one example, it is suppose a grace of a material, and comparatively with that there is a potential barrier. As I told, one CO gas will interact, it will convert in CO2 and release the electron, then the potential barrier will be reduced. So it is an example of n-type material. This is a resistance in air. Once the gas is interacted here, the resistance will change, and after some time it will get saturated. And at that point, if we taken out the gas or gas is off, the resistance will again restart recovering and reach to the initial position. So that is the dynamic curve or sensor response. Or to calculate the response time, which is the total change. So 90% of resistance change, how much time it takes, that is response time. And after removing the gas, the 90% recovery, how much time it takes, that is defined as a recovery time. And similarly, if it is the p-type material, for reduced gas, as I told, the resistance will increase, and after saturation it will decrease. So it can increase or decrease, but the importance is the how much resistance is changing. So it is measure the change of resistance. So it is a typical structure of metal oxide gas sensor. That is very simple. It has few component that is the mostly we use alumina substrate, glass or silicon for that. But most of the sensor they use as an element of alumina, and to provide some elevated temperature, micro heater is fabricated on the back side. Then we use the material as a sensing layer, which will as a work as a detector material, and then over we fabricate the electrodes to measure the sensor response. So it is a schematic diagram of the actual system. We have two probe systems where we introduce the air gas, synthetic air, and the target gas, which we are measuring. So what is the same composition outside, that composition we generate to take as a reference, and that is the actual picture of the gas sensing system, where we have that two probe, and then with the help of that the sample is pasted here, and then we measure that through the Keithley, the actual dynamic curve of the gas sensors. Okay, so now we will discuss what technique we are using for that. We are using the magneton sputtering technique. It is a very renowned technique, and it is the versatile technique which is used to deposit the material of all kind of material. We can do oxide, metals, nitrides, 2D materials, composite, alloy, any kind of material we can deposit. And there are several parameters which can control the material's properties. And the working principle of sputtering is very simple. We introduce the argon gas, then apply high potential, then argon ions are generated, which is generated the plasma, and which is hit the target material, and the film is deposited. So principle is simple, that it is plasma based, but we have the multiple of control parameters there. We can we can deposit the film at room temperature also, at high temperature also. We can vary the working pressure, temperature, power, distance at different angles to tune the material properties. Or it can be used for both kind of material, max oxide, non-oxide, magnetic material, non-magnetic material. So mostly non-magnetic material, we use the balanced magnetron, in which the plasma is confined near the target. And for magnetic material, we use unbalanced magneton, where the plasma is extended to the over the substrate also. And it can be used in the different mode, like based on the, if we are conducting material mostly, then we use DC sputtering, or if the material is semi-conducting or insulating, then we use the RF sputtering. It can be used as a reactive, whatever the material. If we want to the same film as a deposit, that is called non-reactive. And if you want to use from the metal target to oxide, nitride, that is called reactive magnetron. And as I told you, for a non-magnetic material balance, or for magnetic material, we use the unbalanced magnetron. Okay, so now we will discuss what research we have done for on metal oxide gas sensors. So for example, we have deposited a tin oxide film, and to enhance their properties, we have done the surface functionalization but PD material. So these are the basic properties of the material after depositing the film, how the grains are looks like, and in what form the material is deposited, and how defects are generated. If you see there is a, that is the typical spectra of pure metal oxide, are just a few nanometer layer of PPD. It is generated a huge defect on the surface, so it will boost up the gas sensing performance. So as I told, at room temperature, normally the metals have oxide have high resistance. So to reduce the temperature, we increase some the temperature so that resistance will decrease. So at one particular temperature, it will show their maximum efficiency. So when we measure from 100 degree to 300 degree, it is showing at 200 the response, which I told ki, response in air and response in gas. It is showing the maximum response. This is a pure material. And even at 100 degree also, it was showing around 15%. But just a 5 nanometer on that material of 300 nanometer of oxide layer, that is boost even at 100 degree, it is showing over 90% sensor response of PD oxide film, and the response also very sharp. So the time it is coming at 200, about 8 seconds of the response time and 15.3 seconds of the recovery time. And then we have tested how lower concentration it can measure. So from 91 ppm to it is able to measure 2 ppm. So that means the threshold limit which I shown for CO is 35 ppm. But with this sensor, we can measure up to 2 ppm. So that means it has the high sensitivity also. And we have tested it over a period of 6 months. So in a period of 6 months, the response is almost same. So that means it has the high stability also. As I told you, why after few nanometer of PD layer boost up their sensing property, because PD is used as a catalyst, and it is a very good oxygen dissociation. So it catches that oxygen, convert in the oxygen ions, and I told you the total role on the surface is how much oxygen ions are absorbed on the surface, that is reacted with the gas. So PD is increase the availability of oxygen ions on the surface. That why the response is very sharp. So response is increased. So these are the parameter optimum temperature at which it is showing the best response is 200. Sensor response is above 99%, and response recovery time is also very fast, 8 seconds and about 15 seconds. Similarly, as I told you, the preparation of material principle is same of the gas sensor, but the main research is how we can tune the material's properties. So earlier, the last film which I shown, we have used the 10% oxygen during the preparation, and in that case, we have used the 5% oxygen, and then material properties totally changed. That means it is not completely formed as a tin oxide. There is a composition of tin oxide and tin ash. So that means we got the very surprising result when we are measuring, as I told you, N-type material, the resistance should decrease. So resistance should decrease, but initially the resistance was increasing. Multiple times we have used why resistance increase, it is an N-type material. So when we have changed, okay, we will scan the temperature range. So after scanning the temperature, we found that up to certain temperature 225, it is behaving as a P-type material, and after that it convert into N-type material. Then we done the material characterization, so we found that is a combination of SNO2 and SNO, and SNO is a P-type material, or SNO is a N-type material. So there is an oxygen deficiency in one material. It is generated as a PN type, both material in one go. In one material, both type of materials in 2020 it was showing P-type behavior, N-type material. So it can be used as a single material, PN switching also, where we use the two type of materials. In both range, now we were able to measure even less than 1 ppm. It is showing, though it is very less, but still it is detectable 500 ppb. So that is also we have tested over 4 months. So still the dynamic response curve, it is showing almost same. That means it has the high stability also, as I told, that is selectivity is showing the sensor response for CO in comparison to the other gases. So it is with the high sensitivity selectivity also. Similarly, we have used the other material, zinc oxide PD functionalized, that is also for CO gas. So in that case also we have got very good results for both gases. Even in that case also it was able to measure 1 ppm, and this sample we have measured or almost over a 10 months. So after 10 months also it was showing a very good response. That means ki they have the long stability. And as I told ki PD is a very good catalyzer. So pure film is showing the maximum response at 400 degree, and after PD the temperature is reduced to 250, and the response recovery time is also very fast in both case, 8 seconds and 6 seconds. And detection limit, though it is the measurable limit which we have measured, but if we calculate the theoretical limit ki how lowest it can go, there is some calculation based on that. It can measure up to 230 ppb, because there is a system limit which we cannot go that limit, but it is suggested that it can go in ppb also. Okay, so this is the another material one, that oxide. Now it is for the NOx, NO2 or NO. So for that tungsten oxide we have selected is a very suitable material. As I told, if you tune the process parameters, we can tune the material. That is the beauty of thin films. In bulk has the certain limitation, we can tune up to certain limit, but in form of thin film, by controlling the various parameters, we can tune their physical properties in a wide range. So in that case, we were able to measure this sensor work as a NOx, that means for both NO2 and NO gas. At 150 degrees centigrade, it is measuring NO, and with the, this is response recovery time, what we have calculated for the different samples. This is the thickness of the sample, that is also play a crucial role. Even just changing the thickness with keeping all the same parameters, that is also showing the different result. That means ki thickness is also a crucial parameter, because when the gases interact over the surface, up to what depth it is changing the resistance. See, so for that thickness is also need to measure, and also it is showing the NO2. That means is this single material depending on the operator temperature. If you operate it as a 150 degree temperature, it will measure NO gas compared to the other gases. And if you keep that temperature 250, it will measure NO2 gas. That means ki temperature dependent selectivity. Same material can use for both gases NO and NO2 if we keep the separate temperature. As I told you, different climatic condition has a huge impact on that. If you see the impact of humidity, so it was the material when the 36% humidity was there, and when we increase the humidity up to 90%, so it is drastically the sensor response is changed. So that's why we need to calibrate this sensor in the different humidity, different temperature, different climatic conditions to check their performance. That is the third material which on we are working, that PD coated nickel oxide for H2S gas sensing. So this material is highly suitable for that. So that is also we have measured the pure sensing. It is showing very good sensor response at different operating temperature, and with very fast response and recovery time. And also it was able to measure the limit up to 2 ppm, where H2S gas threshold limit is 10 ppm. So it can measure less than that, and it is also highly selective. The same material which was showing other one oxide, NOx, and that nickel oxide is a p-type material which is highly suitable for H2S gas, and that is the repeating current. For that also we got very good response. So in that case we get the optimum temperature at 400 degrees centigrade. So in that case also, when we use the PD coating on that film, the optimum temperature is reduced to 250, and the response recovery time also improved, and that we got high stability also. So this is the, as I told, the initially the schematic diagram of the device structure. So this is for that also we need the micro heater. As I told, ki the oxide sensor, it will work at some elevated temperature like 100, 150, 200. So for that, first we have developed the micro heaters, and then tested it up to 600 or higher temperature, and then we deposit the materials, and over that we have fabricated the interdigited electrode. So it is a multiple device. One device is a one sensor. So it is 64 devices and 64 electrode at the same time. We can deposit over there, and it is after connecting this wires, it is the complete fabricated sensor devices. So summary is that, as I told, the gas sensor testing system is essentially required for the sensor testing and certification to provide services to the commercial sensor manufacturer and sensor developer at academic and research institute, provide a platform in India that will cater to sensor manufacturer requirement for quality assurance for their product in the global market, help to ensure the quality and reliability of air quality data. And then despite the availability of wide variety of sensing material, metal oxide sensors are based on thin films are promising to monitor and detect the various air pollutant gases. Magneton sputtering technique is used for the deposition of PD functionalized metal oxide for carbon monoxide gases, NOx, or hydrogen sulfide. So in case of metal oxide is highly depend on the material's properties. So process parameters like power, pressure, temperature, process gases are able to tune the control of the properties of their qualities, morphology, composition, thickness, which will optimize to enhance the performance of gas sensors. Okay, so I would like to highly acknowledge the thankful to the director, CSIR National Physical Laboratory, our head, Dr. Sachidanand Ji, scientist Ji, team members, Dr. Govind and Dr. M. Senthil Kumar, our collaborator, Professor Davinder Kaur and Professor Ramesh Chandra from IIT Roorkee, and the other faculties, Dr. Jyoti Jaiswal, Dr. Ashwini Kumar, Dr. Amit Senga, Dr. Praveer Pal from CGCRI, and the research group, Dr. Charu Dwivedi, DST Women Scientist, Dr. Dasari Sunil Gavaskar, CSIR RA, Dr. Amit Kumar Gambar, he recently completed the PhD, is doing the postdoc at University of Chile. And Dr. Stuti Srivastav, she has also completed PhD on H2S sensor. Amit has completed on CO sensor. She is also doing postdoc at Minching University, Taiwan. Dr. Rahul Godiwal and these students are currently working, Dr. Abhijit, Mr. Abhijit and Mr. Akash Kumar. And thank you for your kind attention.
Thank you so much, sir. Any queries from participant side?
Very good morning, sir. It's a very nice talk on gas sensor. Sir, I have a very basic question. If I know the band gap, if I know the band gap of a material, so can I get any idea about the sensing properties?
Yes.
There is some correlation of optical band gap with the sensing properties.
Yes, there is a correlation with that. As I told, if the material is highly insulating, band gap is high, it will work at higher temperature. Like in my case, whatever I get high temperature, they are working on 400 degree or 300 degree. If the material is band gap is low, that means ki resistance if in kilo ohm, that means ki band gap is nearby 3 or below. It will more suitable for gas sensing at lower operating temperature. If band gap is high, that means it will work at high temperature. So that means you need to give the high power consumption.
That means if I do any doubling and then reduce the band gap, that means that sample is very good for the gas.
Very good for the sensing. Or the perfect stoichiometry, perfect material is not required for gas sensing. For optical sensing and other application, we reduce the defects, minimize the defect. But in that case, if you deliberate generate the defects, more oxygens are there, material surface is rough. If you generate the porous surface, so that means gas has the more time to interact on the active sites. So that is the same material for different application. You need to tune the properties or band gap also.
One more question, sir, if you allow.
No problem.
Sir, let us consider I have a metal oxide thin film deposited by any technique, like a sputtering and opting. Then what the technique are you used for the electrodes making, sir?
For all the techniques, I am using sputtering. For micro heater, electrode, sensing layer, everything by the sputtering technique.
Thank you so much.
Because it is a repeat, as I told you, once you optimize the process parameter, that is the repeatable technique. In some cases, if you prepare the nanostructure by other metals, sol gel, dip coating, so reproducibility and getting the multiple sample with the same quality is very difficult. But in that case, once you optimize that technique, you will more than 95% you will get the repeatable results. So that is commercially viable technique.
Any other? Thank you so much.
Okay, thank you. Namaskar, sir.
Thank you so much, sir, for your insightful and valuable presentation. Now we move on to the memento presentation ceremony. As a token of our deep respect, we would like to express our heartfelt gratitude. I may now request Dr. Shashiraj sir and Dean sir and Amit sir to kindly present the memento to Professor Dr. Preetam Singh sir. Thank you so much, sir. It is our privilege to now invite Dr. Amit Kumar sir and Dr. Dhirpal sir to kindly come forward and felicitate our esteemed speaker, Dr. Pravindra Kumar, Professor, Department of Bioscience and Bioengineering, IIT Roorkee, with a green planter as a symbol of respect and gratitude. Thank you so much, sir. Continuing with the session, we now invite our next distinguished speaker. It is our great honor to introduce Dr. Pravindra Kumar, sir, a distinguished Indian scientist and professor in the Department of Biosciences and Bioengineering at the Indian Institute of Technology, IIT Roorkee. Sir completed his PhD in Biophysics, Structural Biology from AIIMS, New Delhi. Sir's research expertise span across structural biology, protein-protein interaction, enzyme engineering, and structure-based drug designs. Sir's work particularly focuses on enzymes involved in biodegradation of toxic compounds and plastics, contributing significantly to environmental sustainability and biotechnological innovation. Sir has made remarkable contribution through numerous high-impact research publications and has played a vital role in advancing the field of biocatalysis and environmental biotechnology. In recognition of his outstanding scientific contributions, Sir has been honored with prestigious awards, including the National Bioscience Award for Career Development. Sir continues to contribute significantly to academic research and scientific innovation in India. Sir will be delivering his talk on structural insight into a thermostable hydrolase for phthalate detoxification and PET plastic biodegradation. So we warmly and respectfully invite you to kindly proceed with your presentation.
Thank you, sir.
Thank you. So, can I put it here or this will work? This should work like this. I think, but it's on. Yeah. Thank you. Thank you, Dr. Ravindra Jain, Dean. I think voice is not in this proper. Yeah. Nita, I can. Okay, I can use this one. Yeah. So thank you, Professor Jain and Professor Ashwini, for inviting me here, and thank you all. I would try to tell, in a brief way, what we are doing. So in IIT Roorkee, I want to give a little bit history of IIT Roorkee. So Roorkee was, initially, I don't know how many of you know that Roorkee, how this university came up. So in, I think, 1847, prior to that, there were a lot of problems when Britishers were here. I think there is a, can I use that one? Yeah, because that will be. So when Britishers were here, so that time there were a lot of problems, and India was struggling with Britishers, and Britishers were struggling with us. So that time they thought that what to do. There was a one engineer. So Roorkee was a small town, and he said like, if we can do something for farmers and for these people in North India. So North province was like all Punjab, Haryana, UP, and all. So there was a governor for this thing, Thompson. So he talked to Thompson, Sir Kotle. Sir Kotle was engineer, and they had the army cantonment in Roorkee that time, in a small place. So they said like, if we can make a canal from Ganges, river, so that would be helpful for farming as well as for drinking water. So that's how they started this work. In 5 years, they finished that canal from Roorkee to, not Roorkee, Haridwar to Kanpur, which is about 550 kilometers. And then know how to maintain that. So they wanted to open some engineering college. So they started this Thompson Engineering College. So in 1847, more than 178 years ago, those guys, they started this canal. And then if you see that, when the canal, I think long back, this picture might have taken. So this looks like, from Roorkee, you can see this canal and then foothills of Himalayas. This is like, all Himalayas you can see. So it was a beautiful canal, and one can see that, it was useful for us. So some of the work they have done nicely. But now what we have done, look at this now. So we have this sort of rivers around, and then we are dumping all sort of waste in this. And then that waste, somehow it comes to us, whether it's through water or through soil or by some other means. So these sort of things we are doing. And if you see that, we are also putting a lot of pesticides in our, agriculture land. Also, most of earlier females, now even males, they are putting a lot of cosmetics. We use shampoo, conditioners every day. They are all toxics. They are made up of plasticizers, which are carcinogens. And then cosmetics, even nail polish, lipsticks, whatever, females are using, they all are toxics and they are all carcinogens. And then plastic bottles, most of us drink water in the plastic bottles. So they also, like, after some temperature, these plasticizers leach out into water, which is carcinogen. So what I am trying to tell you, that may not be science what we are doing, you may not be able to understand, but at least you can prevent using these things if possible. These days, I think most of us started using conditioner and shampoo every day, but we don't know how much we are absorbing. So it would be at least this much you can do, something natural thing you can use rather than using all these things. So these were some of the compounds, toxic compounds, which are being utilized in a lot of industries, and we have used enzymes to degrade this thing in last 20 years. 21 years I am in IIT Roorkee. 2005 I became a faculty there. So I was told that no, I should not go into the science details, but I'll try to tell you a little bit. So there is a pathway of enzymes which are required for degradation of toxic aromatic compounds, and we engineer those enzymes later on so that it can become more robust and we can do things in a better way. So understand that now if we have seizures and if we want to cut something, so seizures is an enzyme. Or you can see lock and key. Key is the the toxic things, and lock is an enzyme. And if we can make lock in a way that a lot of keys can be fitted, that means a lot of toxic compound can be fitted in that. So that's what we are doing in enzyme engineering. So you can say कि जो ताला ठीक करता है वो हम हैं. So that way we try to engineer enzymes so that a lot of things can be added into this. So what happens if you see NADH is a small compound when you use bacteria, so that can produce, otherwise you can put outside from this, and then electron can be transferred to this NADH to the reductase, and then from this to pyridoxine. And there is an enzyme called BPDO, biphenyl dioxinase. In this, this electron comes here, there is a ferrous ion center. So this, near this, there is a dioxygen will bind, and dioxygen is inert. It activates that thing and that gets inserted into the aromatic ring. This aromatic compound is quite inert, but if you insert oxygen, then it becomes soluble. Then that can be degraded. So this enzyme, the entire thing does this thing. So just to tell you that we do crystal structure, we make for these enzymes like a sugar particle, this small crystals, or females, they like crystal in different form. Diamond is also crystal, so we make like diamonds from protein or enzymes. So then we bombard X-ray on it, and then we look at the three-dimensional structure of those proteins like here. And this is the overall architecture. We find out that where is the iron, and also not going into much details. But if you see this in cyan color, this is how the biphenyl was bound. So aromatic compound was bound in this. So this was the key for our lock. So this we can see here. If you see that different bacteria, so bacteria grow on these kind of things slowly. But if we know which enzyme is responsible for degradation of that, then we go in details of this thing. If you see the magenta color here, pink you can call it, and arrow is given here, that which is the entry to this active site where these aromatic compounds can be fitted. So this is B356 name we have given to this bacteria, the LB400, where there are two entrances. So if you have broader this molecule, then we require LB400. If you have narrower, then we require these things. So when I showed biphenyl, there are two benzene rings were fused together. You can have chlorine at ortho meta para positions. So, and if you put ortho meta para positions chlorines and different permutations combinations, you can have 209 different compounds, and they are being utilized for industry. I am not going in for details, but they are being utilized. They are all carcinogens. They all cause cancers. So what we are trying to do is we try to see which bacterial enzyme is better for degrading more number of compounds. So by looking at these structures, we can change these enzymes things. We compare these structures and then see which are amino acids are nearby, and these amino acids we mutate. We do site-directed mutagenesis by looking at this structure. So I don't know how many of you know this thing, but that can be changed by doing this thing. And by doing this, we can engineer this. So this is how we have done. And so if you try to compare those active sites so that they block each other, so we try to cut this part also from here. I said from seizures, so we can change that thing. So that's engineering of that enzyme. So first thing was dumping things in the canal. Now these are the plastic bottles, which you see that all are lying this. So then we wanted to find new enzymes which are, so we go to these sites and we find the bacteria, we isolate those bacteria, and then we see that what are the problems. So they cause all sort of cancer, infertility, obesity, diabetes, many different disease if we inhale those sort of things. And so we isolated different bacteria which are mentioned. They can grow in this thing, but very slow. They might take maybe millions of years to do this thing. By the time our many generations will pass. So we need to do faster things. So we try to utilize these pathways. So there are different enzymes are involved in this, where right signs are there, so that those are enzymes. So this is this polymer of phthalates, and then first enzyme esterase remove this chain, then another one remove other chain, then this dioxinase put hydroxyl group. This is how eventually this makes protocachuate. And protocachuate can be degraded by any kind of bacteria. So what we do, just flow I am telling you, what we do, not going in much details. So we do clone that particular gene of that enzymes. Then we see the expression of that gene and purify that thing. Here, if you see, then look at the purification, profile. Then make those crystals, which I was telling diamonds, they are sugar particle of things. Then bombard X-ray. From this, using Fourier transformation equations, like we use maths also, and computers both, then we try to solve the structure, three-dimensional structure, and then we see that where this component of, this is terephthalate, the one molecule of these plastic bottles which you drink. So the polymer of this makes the plastics. So how it binds. And then eventually, by doing site-directed mutagenesis, we make the enzymes or bacteria engineered, and then we look at other things. So this is what we've been doing. So those people who want to take interest, they can do this thing. So by doing this, so then we use LCMS. We can see that by these techniques, whether this is being degraded or not, these compounds. And when you see this from the active side, we can see how they enter, how they go out. So all these details, we can see that. And whenever you see this density sort of thing, this covering net, that's the electron density map comes out from X-ray diffraction. So we can see how this is bound and all. So as I was told that no, I should not go in more details, just I'm telling the overall movie of this thing, that know how it enters these all plasticizers and where from it goes out. So, and eventually this is the inside structure of this. So this is entire things we can know about that enzyme. So this is enzyme, how it is working. So you can see the real way of, how enzyme overall it looks like. All right. So now that second enzyme, similarly, we purified, crystallized, and then we have seen the activity of that enzyme against this phthalate, which does the way, biphenyl dioxinase were doing. So all proteins, they are in dynamic state. So what you are seeing moving, they are moving in the cell. So you can see their movie is completely, this is a tetramer sort of thing. And in this also, this phthalate dioxinase things we can see, and then eventually, this is the compound which is bound here. So we can find out the position, active site where those compounds are. And then by looking at the position, we can, change those amino acid. And then that is the engineering of this thing. So this was for phthalate dioxinase. And so we go in more details, but, not getting, if you have any questions, so we can tell later in more details. So this is the complex eventually we have made for pediophthalate complex. And so here when you see this blue density, that means this has come in real crystal structure, this complex. Then we had made this terephthalate. This is, I told you, this is the plastic bottles which you utilize it. And we see the comparison with other enzymes, and by looking at those comparisons, we try to do the mutagenesis further. All right, so this is the first thing. Then when we were doing this thing in Japan, they published in Science that they found out new bacteria which is doing the degradation of these plastics. And a lot of work was shown that 60 milligram PET film was degraded in 20 days. Then another work was that 60 milligram was done in 10 days, and now finally 2 days. But in our lab, we can do in 6 hours same thing by our enzymes. So this is what all PET biodegradation work was done, and we did the crystal structure of that also. We cloned, we purified the enzymes responsible for this, and we did the activity. And so we got the crystals, beautiful crystals, diffraction, and then crystal structure. And from this, we could see that how this particular structure looks like. So by doing all these things, we can again see that, how this terephthalate degradation enzyme looks like. So all details can be seen from here, and then you can see these surrounding amino acids. By looking at this thing, you want different kind of compounds to be fitted in, so we can change these things if you know the structures. And we do mutational studies, and we see that which particular mutation is better. We do the comparison with other enzymes and then see the differences, and then where we see the differences, we can make the mutations there. So if you see that in biphenyl dioxinase that there are changes here, so we can look at those changes and do the changes. So eventually, so this is whole idea of our lab, that so if you see these things, they are making these plastic bottles, you are making household waste, and make this waste of mountains here, and then you generate this sort of kind of pollution. Whereas we try to do from this, we make these enzymes engineered, and we try to make them, utilize and make usable compounds from this, commercial compounds. From this, we can make the bioplastics as well as biofuel. So this is what we are trying to do. So there are more than 1 or 2 enzymes. So we look at all these enzymes, and we can do the mutagenesis of these enzymes here, whatever the changes you want, we can then do the immobilization of those bacteria or enzymes, and eventually we celebrate our holidays, and then we enjoy, this is how our team is working. And I'm with you if you have any question. Thank you so much. Yeah. So I was told that I should not go in more details by Ashwini. So I just showed the things, but I will be happy to answer any question if you have question, because interaction is more better, I think, rather showing data. Anybody has any question? Yes, please. So I think somebody gives her mic, and then I can answer from here if I can hear it properly.
How does the temperature affect the rate of reaction?
Yeah, good question. That that we have to see. So we screen a lot of bacteria, and then we see that which particular bacterial enzyme is thermostable, and we by screening we find out first of all from wild type, but then by looking at the structure we see that which sort of amino acid could be responsible. So we keep on changing those amino acid and keep on checking that which is most thermostable, that thing. So that way, so depending on we need to change that activity of that enzyme. Some require like at room temperature. And for example, if you want to degrade plastics, so it's always better to degrade it at room temperature, because you don't waste energy. So we don't want at high temperature, but in general, whatever wild type enzymes are, they are more stable at 60 degree or 45 degree for degradation point of view. Yeah.
Sir, my question is, how can I join your team?
That is good question. So you should come and qualify whatever requirement for IIT Roorkee, and give my choice. Will be happy to take you. So simple way is, there are students who apply there, and if they are eligible, then they apply. Then we select them for interview, and then if we think that the student is capable of doing PhD, then we take them. And then if they give, it's mutual. If they give my name first, and then I gave their choice first by interacting, then this is how they are selected.
So right now I'm doing PhD from this university.
Yeah. So so then you continue doing this thing. You can learn things. You don't have to do PhD. With the person only. Then you can communicate with different ways.
Your work is fascinating.
Thank you so much. You can interact with me without doing PhD also. Okay.
Thank you, sir.
Anyone else has any question?
Any other question from the gathering, please?
For the pharmacy,
Yeah.
So that was some related to the pharmacy person.
Yeah.
I want to ask, whenever we are going to change out bacterial,
I can hear you. Yeah.
So whenever they will convert the metabolites, which is always that whenever bacterial enzymatic kinetics, they will convert the metabolite. They are all are the non-toxic.
No, it's not like that. So many times there is whatever they converts. So we have to see what are the different metabolites they are converting. Sometimes they become bottleneck for next enzyme. So there's a cascades of enzymes. It's not one enzyme which is working. There are several enzymes which work in this cascade method. One first will produce some product, and then that will be substrate for another one. But that metabolite sometimes becomes toxic for next enzyme. So we need to change that. That's how so I didn't go in more details, but yeah, we need to modify enzymes for doing that toxic thing also we remove. Yeah. So anyone else? I thought there will be more questions because I have talked about cosmetics also, that many females will question me like, why, what they should use it. So at least if not science. So anyone use sugar cosmetics here? No one use sugar cosmetics. So sugar cosmetic girl Vinita Singh, she was a PhD, my PhD supervisor's daughter. So she was telling me when I was presenting once, so you are trying to spoil my business. So I said, better not to use plasticizers, use, better. So I think she's using better things now. So this is what I heard from her. So there are people like, I'm not, advertising for her, but that's what she said. Like,
She said that she formulated another way.
yeah. So she was a small kid when I was doing PhD with her father, and she was in 7th grade. But like, we interact, and then I came to know that she doesn't use plasticizers, but very expensive products she's selling in the market. Yeah. Yeah. Okay, then. Thank you so much. If no interest.
Thank you so much, sir, for such a wonderful and informative sessions. Now, we move forward to the memento preparation ceremony. As a token of our deep respect, we would like to express our heartfelt gratitude. I may now request Dr. Amit Kumar sir and Dr. Lubhan sir to kindly come on stage and facilitate our respected esteemed speaker, Dr. Pravendra Kumar sir, with the memento. Now it's time to honor our session chair. And to honor our session chair, Dr. Lubhan sir, I would like to call Dr. Neeru ma'am and Dr. Sarita ma'am to kindly come on stage and present the memento to our session chair. Now it's time to facilitate our reporter of the session, Dr. Ashwini Kumar sir, HOD Life Sciences. For that, I would like to call Dr. Dheerpal sir to kindly come on stage and facilitate our reporter of the session. We now proceed towards session 9, poster presentation, where innovative ideas and research finding will be showcased. These presentations reflect the creativity, dedication, and scientific curiosity of our young researchers. And to judge our poster presentation, we are honored to have our judges, Dr. Aniruddha Srivastava sir, Associate Professor Chemistry, Dr. Vikas Kumar sir, HOD Basic Sciences, and Dr. Naveen Chandra sir, Associate Professor Computer Application. For that, we request all the offline poster presenters to kindly move towards their respective venue for the poster presentation at Science College. नहीं चलेंगे ना. सर आपकी पेन ड्राइव. किसकी है? वो वाली. ऑनलाइन वालों को ऐड करो.
तो एक ग्रुप पे मैसेज डालो या इसको बोल दो ये तो लाइव है ये लाइव है
पर आप इसमें कर
पहले
और जो है
ग्रुप पे डालो
मेरे पास एक्सेस नहीं है ग्रुप पे डालने का. ओनली एडमिन कैन सेंड मैसेज एंड आई एम नॉट एडमिन.
Ritul, Ritul. Next we have Aditi Chauhan. Aditi Chauhan. Divyanshi Giri.
Yes, ma'am.
Giri. Kindly share your poster.
ओके मैम.
Ma'am, my slide is visible or not?
Yes, it is visible. Start presenting.
Okay, ma'am. So, good morning, everyone. I am Divyanshi Giri. I am MSc student and currently studying MSc Physics from Gurukul Kangri University. My topic is optimization of Fe doping in V2O5 nanoparticles for improved structural and optical properties. So basically, I have worked on V2O5. My abstract is this project present the effect of Fe doping, basically 5% and 10% on the structural, optical, and morphological properties of V2O5. V2O5 is widely used in photocatalysis and gas sensing, but it suffers from high charge recombination. The objective of my work is to study how 5% and 10% Fe doping affect structural, optical, and morphological properties. So we have synthesized with 205 particles here with sol gel method and characterized it using XRD, PL, and FESEM techniques. This is the XRD graph. From XRD analysis, we confirmed the orthorhombic structure of V2O5. After doping, the crystalline size decreases and lattice strain is introduced. The PL graph shows a decrease in emission intensity, which indicates reduced electron hole recombination and improved charge separation. FESEM image reveals a more uniform and improved structure after the doping. So, after comparing, 5% doping is optimal. It enhances crystallinity, optical performance, while 10% doping causes excess lattice distortion. So, in conclusion, I would like to say that controlled Fe doping, especially at 5%, significantly enhances the properties of V2O5, making it more suitable for photocatalytic applications. Thank you.
Thank you, Divyanshi. Questions?
Only for this.
Hello, Vivekanand, you can hear me? Hello? Hello, yes, ma'am. Can you please tell me by which property, from which property you can say that your 5% doping sample is best for the photometallic application? Please justify it. Ma'am, due to characterization techniques, on the basis of characterization techniques, I can explain the photocatalytic activities. From which characterization technique? Structural, optical, by ma'am. Yes. No, optical properties can be explained by PL analysis. Okay. Yes, ma'am. Okay. Thank you. Okay, thank you.
Thank you, Divyanshi. Next, we have Janvi Kukreti. All the participants are requested to kindly open their camera and mic while presenting. Janvi Kukreti. Janvi. Unmute yourself. Janvi. Nikita Pundir.
Yes,
Nikita
ma'am.
Pundir. You can start presenting. Kindly open your camera and mic.
कहाँ है?
Nikita, kindly share your screen.
मैम
Okay, it's visible.
Yes, ma'am.
Open your camera and start presenting.
Good morning, everyone. My name is Nikita, and today I am presenting my work on the effect of tungsten doping on vanadium pentaoxide nanoparticles for thermoelectric applications. This work presents the V-doped V2O5 nanoparticle with 3% and 5% concentration with synthesized to study their potential for thermoelectric applications. Ma'am, X-ray diffractions analysis confirmed orthombic V2O5 with a reduction in crystallite size after W doping, indicating successful incompression and lattice strand. PL results show decreased emissions intensity in doped sample, suggestion reduced charge carrier recombinations. Morphology studies reveal a decrease in grain size compared to pristine V2O5. The main objective of this work is to synthesize V-doped V2O5 nanoparticles to study the structural change in using XRD. The XRD patterns confirm that formation of orthombic V2O5 structure in all samples, including pristine 3% and 5% W doped sample. The main peaks corresponding to plane such as 001, 110, 400, and 002 are clearly observed, indicating good phases purity. The pristine samples show that sharp and intensity peaks, which indicates high crystalline. In 3%, V-doped sample peak boarding is observed, suggesting reduced crystallite size and increasing lattice strands. In the 5% V-doped sample, the peaks becomes observed with doping, which confirms that successful incompression of tungsten into V2 or 5 lattice. The PL analysis showed that the emission intensity decreased with V-doping. The decrease indicates reduced recombinations of charge carriers, which means better charge separations. This is beneficial for improving the efficiency of thermoelectric and other functional applications. हम इसका डेली यूज कर सकते हैं as waste heat recovery from vehicles and machines, power generation from body heat and wearable devices, and cooling applications like small electronical cooling system. This application helps in energy saving and sustainable technologies. In conclusion, we can say V-doped V2O5 nanoparticles were successfully synthesized with orthombic structure and high phase purity. XRD results show that doping causes lattice distortion and change in crystalline, while PL results indicates reduced
Nikita?
charge recombination. Yes, ma'am.
Your time is over.
Okay, ma'am.
Question.
Nikita? Hello, Nikita?
Yes, sir.
You have prepared a vanadium oxide nanoparticle, is it?
Yes. Yes, sir.
So, what is the procedure you have adopted? Because here you have shown only the XRD.
Sir, it is salt gel method we use for preparing this V2O5 nanoparticles.
And how? What is the size of the nanoparticles? And how you gonna confirm that? Did you measure the particle size?
No, sir, I will study on this.
So without size, how can you say that you have prepared a nanoparticles?
Sorry, sir.
It's just you can query, okay, because you just have given us XRD. XRD it will give a, because you have a using a vanadium also, okay. So maybe it will give the vanadium peaks, okay. So only the XRD peaks. It's not confirmed that you have prepared nanoparticles. You have to go for a particles DLS measurements and as well as also a SAM or a time level measurements. Okay, so that time you will confirm that you have a nanoparticles.
Yes, sir.
Okay, thank you.
Thank you, sir.
Thank you, Nikita. Kanishka, Shakibul Hassan, Ritul, Ritul, Aditi Chauhan, Aditi, kindly unmute yourself.
Yes, ma'am. Am I audible?
Yes, you are audible. Kindly share your presentation.
Good morning, everyone. I am Aditi Chauhan. I am currently pursuing MSc Physics from Gurukul Kangri University. So here the topic of my poster is structural and magnetic properties of cerium oxide nanoparticles synthesized by Sol gel method. The abstract of my poster presentation is Ceria nanoparticles of average size of 4 to 6 nanometer are synthesized using conventional Sol gel method. The prepared compositions of cerium LaO2 are investigated using XRD, FTIR, and magnetic measurements. Ritual refinement analysis confirmed the formation of FCC structure corresponding to CeO2 with space group LA doping into CeO2 nanoparticles results in decrease of average crystallite size. In addition, FTIR results further confirm the phase of samples for CeO2. However, the magnetic properties of these samples are found to be enhanced after LA doping. In the next section, I would like to explain the introduction part of cerium oxide, where I explain the some basic properties of cerium oxide, such as cerium is the second member and the most reactive element in the lanthanide series. Being electropositive in nature, cerium exists in dual oxidation modes, Ce3 plus and Ce4 plus respectively. And the next one, cerium oxide is insoluble in water. It's a rare earth element, boiling point having 3500 degrees Celsius. Some other basic parameters such as crystallic structure has having cubic fluoride, space group FM3M, band gap 3.3 electron volt, and color pale yellow white, density 7.65 gram per centimeter cube. Here I done the XRD analysis of cerium oxide doped with lanthanum. Here XRD analysis done by ritual refinement using foolproof software. These are the two graphs I have plotted here versus intensity and 2 theta degrees. In the next part, I have shown the electron density distribution, where in the four corners, the electron this shows the cubic structure, and the next part explains about the Raman spectrum. Raman analysis. Here we observe that in this graph by peak shifting, we can analyze that lanthanum perfectly docked in Syria and it enhanced the some physical and chemical properties of cerium oxide. In the next part here explains the SEM analysis morphology. Here I put the two images which have done. The magnetic properties having pure cerium oxide, mostly non-magnetic, but in bulk form it shows weakly paramagnetic and paramagnetic. After doping with lanthanum, it becomes weakly paramagnetic or paramagnetic. The conclusion of this my work, relaxation mechanism and universal dielectric relaxation UDR model have been fitted to dielectric behavior of the samples. Multiple polarization mechanisms contribute to high dielectric performance over a wide frequency range. Frequency dependent variation in AC conductivity has been discussed on the basis of correlated barrier hopping CBH mechanism. The lanthanum doped cerium oxide nanoparticles typically have an average crystallized size between 3 nm and 20 nm, depending heavily on the synthesis method and calcination temperature. The calcination temperature is done at ranging between 600 to 800 degrees Celsius respectively. Lanthanum doping generally stabilizes the cubic fluoride structure of ceria with higher temperature resulting in larger particles, often in the range 5 to 15 nm range. Some other key size characteristics are as follows, like as synthesized at low temperature, often 3 to 8 nm when dried at lower temperature, higher temperature calcination
Aditi,
grows to 5 to 15 nm after heating to 600 to 800 degrees Celsius due to grain growth. Typical ranges of ceria. Reported
Aditi,
values for acid of ceria generally fall within the 7 to 14 nm range for nanocrystalline powders. Structure. The particles retain a cubic fluoride type structure and adding alid
Aditi,
typically expects the lattice parameter compared to pure ceria. Yes, ma'am.
Aditi, kindly conclude this.
Yes, I would like to I would like to conclude the results of this activity has been discussed on the basis of correlated barrier hopping CBH mechanism, and I had done the um, electric density distribution and XRD analysis. Thank you.
Aditi?
Thank you, Aditi. Questions?
Aditi? Hello, Aditi?
Yes, sir.
You have written there sodium oxide with water insoluble. Is it?
So sorry, voice not audible to me.
You have written there sodium oxide is water insoluble in your poster. No other device. Please switch off your other devices, please.
Sir, your voice is
Aditi,
not audible due to some network issues, maybe.
Aditi, kindly switch off your other devices.
Yes, ma'am.
Aditi, are you, what is? Hello.
Aditi, can you hear our voice?
Yes, ma'am.
Thank you, Aditi. Janvi, Janvi Kukreti, Janvi, Janvi, Ritul, Ritul,
Yes, ma'am.
kindly turn
Am I
off your
audible,
other
ma'am?
devices. Yes, yes, you are audible. Share your poster.
Hello.
Yes, Ritul.
Yes, ma'am. Hello.
Yes, Ritul. Turn on your camera. Share
Yes, ma'am.
your
Am I?
poster. Ritul, Janvi Kukreti, Kanishka, Ritul.
Yes, ma'am. I am audible.
Yes, you are audible. Kindly mute your other
Okay.
devices.
Okay, ma'am. I'm presenting my screen.
Okay.
Ma'am, is their screen visible?
Not yet.
Ma'am, now?
No.
Ma'am, now is visible my screen?
No, it is not visible.
Okay, इसका तो
Janvi,
Ma'am, Janvi Kukreti, am I audible?
yes, you are audible. Share your poster.
Yes, ma'am.
On. Switch on your camera.
Ma'am, is it visible?
Yes. Start presenting.
Ma'am, my name is Janvi Kukreti and I'm MSc Physics student from Kanya Gurukul, and my poster presentation is on Fe dopant tuned the properties of vanadium pentoxide nanoparticles. And my abstract is this study investigates the effect of iron Fe doping on the structural, morphological, and optical properties of vanadium pentoxide V2O5 nanoparticles.
Pure and FE-doped V2O5 samples, 3% and 10%, are analysis using X-ray diffraction XRD, photoluminescence PL, and field emission scanning electron microscopy FESAM techniques. XRD results confirm that FE-doping induces lattice distortions and increases structural disorder while maintaining the orthorhombic V2O5 phase. Photoluminescence analysis reveal reduced charge carrier recombination with Fe incorporation, indicating improved optoelectronic behavior. FESAM shows noticeable modification in surface morphology, and grain size reveals change in V-oxidation states. These findings demonstrates that Fe doping effectively tunes the structural and optical properties of V2O5 for potential application in sensing and optoelectronic devices. And my introduction, which is vanadium pentoxide, I have taken vanadium pentoxide, which is 3% and 10%. And the structure of vanadium pentoxide is orthorhombic layer type. Its band gap ranges from 2.2 to 2.3 electron volt. Its melting point is nearly 690 degrees Celsius. Its boiling point is nearly 1750 degrees Celsius, and color is yellow to reddish brown, and its density is nearly 3.36 gram per centimeter cube. V2O5 is an important material which is used in sensors, electronic devices, and batteries. To improve on this work, Fe dope affects the structural, morphological, and optical properties. Now, this is the techniques of V2O5. First one is XRD, X-ray diffraction technique, and XRD confirms the orthorhombic crystal type structure, and this is the graph for XRD. Second one is photoluminescence PL, and this is the graph as shown. And PL confirms that doping improves the electrical properties of V2O5. And next is FESAM, field emission scanning electron morphology, and these are shown for FESAM. And FESAM shows that doping affects the grain size and surface texture. Now, I would like to conclude the doped nanoparticle exhibit better stability and overall performance. And next is the study confirms that doping is an effective method to tune the properties of nanomaterials for advanced technological applications. X-ray diffraction analysis confirmed the orthorhombic phase of vanadium pentoxide with slight weak shifts indicating successful Fe incorporation into lattice. Thank you, ma'am.
Thank you, Janvi. Questions?
Sir, your voice is not audible.
Janvi?
Yes, sir.
Again, my simple question is: you have wrote down the nanoparticle and confirm the size. Okay. My previously
Yes,
I have
sir.
asked the question.
Yes, sir.
How would you calculate the size or measure the size? Because
Sir,
XRD and SEM is not the accurate method for determining the nanoparticle size.
so for 3% FeDO V2O5, its size was nearly 20 to 50 nanometer, and for 10% FeDO, the size was nearly 10 to 30 nanometer.
But you have seen your FESEM. Okay. The size is very huge, around more than 10 microns.
Hello.
Did you check the SEM image?
Sir, I can't hear you.
You have checked your SEM image.
Yes, sir.
And what is the image pixel size?
Sir, for 3%, it was.
No, I'm just asking because you have confirmed the size from the FES SEM image. See, what if you talking about the nanoparticle? nanoparticle cannot be confirmed from the XRD and the SEM. Okay. What? Hello.
Hello.
Are you hearing me? Hello.
Yes, sir.
So,
Sir, XRD crystalline size is nearly 10 to 30 nanometer.
XRD cannot be confirm the size. XRD give the structure. Thank you.
Sir, it's
It's the crystalline. It provide the crystalline structure. Okay. It's not providing the dimension of the nanoparticle size. Okay.
Sir, dynamic light scattering DLS.
You have done. Have you done?
Sir, I don't have done, but I have an idea.
So you should have done. Keep it here, okay? Because without you cannot tell you have prepared a nanoparticle. Okay.
Okay, sir.
Yeah. Okay. Thank you. Thank you, doctor.
Ritul?
Yes, ma'am. Am I audible?
Yes, you are audible. Kindly share your poster and turn on your camera.
Ma'am, is it screening?
Not yet.
Yes,
Ma'am,
yes.
now?
Start presenting. Yes, yes. Start presenting.
Good afternoon, everyone. Today I will present my work on the influence of lanthanum doping on cerium oxide CO2 for the enhancement of dielectric property. This study focused on improving the electrical properties of cerium oxide nanoparticles using lanthanum doping. First of all, we have to discuss about the introduction of it. Cerium oxide is an important material widely used in catalysts, fuel cells, sensors, electronic devices. It has a cubic fluoride structure and good thermal stability. However, its dielectric properties can be further improved. To achieve this, we introduce lanthanum doping, which creates oxygen vacancies, modifies the electronic structure, and enhances the polarization mechanism. Now we...
Ritul, kindly do your poster.
Okay, ma'am. Ma'am, now is it okay?
Okay.
Okay. Now the main objective of this study, we are to synthesize LA-doped CO2 nanoparticle to analyze structural and vibrational properties and to study dielectric behavior and to understand how LA concentration affects performance. Now we study about the material and method. The nanoparticle was synthesized using the Sol-Gel method. We ensure good homogeneity and control particle size. Characterization techniques are used XRD, X-ray diffraction for structural analysis, Raman spectroscopy for defect and vibrational study, scanning electron microscopy SEM for morphology, and dielectric measurement for electrical properties. Now we have the basic properties of CO2 like structure, crystal structure for like a cubic chloride, space group, FM3M, band gap, 3.3 electron volt. Now we first analyzing the XRD analysis, in which XRD confirm pure cubic phase of CO2, no impurity peaks, successful LA incorporation, peaks slightly shift with doping, lattice modification. Now we study about the Raman analysis, in which a strong Fe2G peaks is observed here with LA doping, peak broadens and slightly shift occurs. This indicates the increase in oxygen vacancies and structural disorder. Now there is a electrical analysis in which they versus in which there are the graph which versus about the temperature. Dielectric constant increase with temperature, higher La concentration, higher dielectric constant, and enhanced polarization due to defect space charge discontribution. And there is a dielectric loss 10 del, which loss peak observed at certain temperature.
peak shift with frequency and relaxation behavior in the material, charge, carrier, hopping mechanism. Basically, the conclusion about it, lanthanum doping effectively enhance dielectric properties. It increasing defect without damaging the structure, improve dielectric performances, make it suitable. Future work, highly doping concentration, thin film application and device fabrication. Thank you.
Thank you, Ritul. Questions? Thank you, Ritul. Thank you, all the participants. We will begin our validatory session soon.